Okay, so doing a Unit 6 test review here. So starting in Chapter 13. Chapter 13 was going over DNA replication. And beginning with comparing it, DNA replication in prokaryotic versus eukaryotic cells. In a prokaryotic cell, you're going to have your origin and replication. Well, first off, back to prokaryotic cells, they have a circular chromosome. So here, in your prokaryotic cell, they would have their origin replication. So that's where you would pull apart that double-strand DNA, and then DNA replication would begin. It would go, you call this the replication fork, where, you think of like a fork in the road. You could go, either that way or that way. So the replication, and they'll call this replication bubble, this whole thing. And it'll proceed in two different directions until you have completed doing DNA replication. So fairly straightforward, fairly simple. Now you compare that to a eukaryotic cell. In a eukaryotic cell, you get multiple replication bubbles. Okay? So compared to just the one single replication bubble we have in a prokaryotic cell, eukaryotic cells, you have multiple bubbles. And so here would be one bubble, and then they would each proceed. So there's a bubble, there's a bubble. They would each proceed until they all meet up with one another. Okay, so there you go. Getting into how DNA replication works. There's a lot going on in this picture. Let's begin with DNA is that double helix. Before we can do anything in replication, we have to use helicase. So that's this protein right here. So we need helicase to unzip the DNA. So this helicase unwinds it. And before I forget it, because I don't have it shown here, you have another protein bonded to the DNA further downstream that we call topoisomerase. Okay, so topoisomerase, its job is to reduce tension. Basically, the idea is like, think of like a long cord that gets tangled up. If helicase keeps unzipping the double-stranded DNA, eventually you would get kinks occurring, like little knots occurring down the chromosome further down there. Topoisomerase, its job is to, like, if a kink starts forming, it'll break that kink and straighten out the DNA so it doesn't bundle up. And then, if we unwind the DNA, we want to make sure the DNA doesn't like re-zip back together. So we have these silverish-looking proteins. These are called single-stranded binding proteins. Okay, and they're abbreviated SSB. So SSB proteins will prevent the DNA double helix from like reattaching back together. Now, DNA polymerase, so this is right here, DNA polymerase, and they call it DNA polymerase 3. So DNA polymerase 3, that's the main enzyme that is actually doing replication. So it's the main thing that's actually adding the bases. Now, DNA polymerase 3 can't just start making a copy of DNA. It needs what we call a primer. So this is an RNA primer. This RNA primer basically is needed to get a DNA polymerase 3 going. Now, the thing that lays down the RNA primer is called primase. So primase adds the RNA primer. So I'll start adding these functions here. Helicase unzips. Topoisomerase untangles. Single-stranded binding protein, that is going to prevent the two strands from going together. So prevent strands, say reattaching, be the quickest way. Prevent strands from reattaching. So then primase lays down the RNA primer. DNA polymerase will actually add the nucleotides. And I want you to note it adds nucleotides to 3' end. Now, 3' end of previous nucleotide. In other words, we grow the DNA strand from 5' to 3'. That's something you got to memorize. We always grow it 5' to 3'. That means the template strand, if we're growing it 5' to 3', that means the template strand would be 3' to 5', relative to where you start adding the DNA nucleotides. So Looking here, there's two strands when we're doing this. There's something called the leading strand, which is this topmost strand, the leading strand, and then what we call the lagging strand. And why we have the presence of a leading and a lagging strand, it all comes back to this idea of DNA having to be grown from a 5' to 3' direction. So we focus on the leading strand. If I unzip the DNA, this would be, for the template strand, this would be 3', this would be 5'. So then, as I said, DNA polymerase has to work from 5' to 3'. So on this topmost strand, if we focus on this kind of zoomed out picture, this topmost strand here, we have no issue going leading strand here. And if you think about the replication bubble, there would also be a leading strand on the other side. So this right here, if I continue this picture on, this over here, this would be also a leading strand. Okay? And then, anyway, so yeah, don't forget there'd be two leading strands because you have a bubble. So then the lagging strand comes about because this other DNA strand, this is the 3' end, and this right here is the 5' end. But the direction that we're trying to replicate the DNA is this way. Like the direction we're trying to go on this strand here is this way, but this way is 3' to 5'. So the issue is, again, DNA polymerase can only go 5' to 3'. So what happens is we have to replicate it in these little fragments. We call these fragments Okazaki fragments. Okay? And and that's what these are here. This would be an Okazaki fragment, that's an Okazaki fragment, that's an Okazaki fragment. Where each Okazaki fragment, if you see that red thing right there, that represents the primer. Each Okazaki fragment has a primer laid down by primase. So what DNA polymerase 3 would do is, we focus here, DNA polymerase 3 would attach where the primer starts. It would copy that fragment until it hits the primer of the lagging strand of the Okazaki fragment it had previously done. So it hits here, DNA polymerase would detach, and then it would backtrack and go to this next Okazaki fragment that was just revealed by helicase unzipping the DNA and primase laying down the primer. DNA polymerase 3 would add the nucleotide to the 3' end until it hits the Okazaki fragment it had just done. And while all that's happening, helicase is unzipping more DNA. Primase is laying down a primer back here, and then DNA polymerase 3 would hit this, come over here to this primer, and then do a DNA replication until it hits this primer, and on you go until it will meet up, until this whole replication bubble is met up with the previous replication bubble, kind of like this here. Okay, so kind of a lot. Not quite done. We have to, all of these fragments, we have to, and same thing goes for the leading strand. RNA, DNA is DNA, right? If you're replicating the DNA, you don't want these RNA primers to remain. So what we're going to need to do is you need DNA polymerase 1. So this is DNA polymerase 1, not 2, not 3, DNA polymerase 1. It is going to remove the RNA, and add DNA, okay, because we don't want that RNA primer to remain. And then this protein right here is DNA ligase. So DNA ligase will glue strands. Specifically, it's going to glue, it'll like, when I say glue strands, it could specifically refer to the Okazaki fragments. Is that still in the camera view? Okay, cool. It would specifically glue the Okazaki fragments together. Or it could also be used, again, it would be used in the leading strand whenever you're just trying to just simply remove a primer. So it's not, in other words, you don't just use ligase just simply to remove Okazaki fragments. Oh, we also are going to use ligase when we do DNA excision repair. I'll show you that slide here in a minute. More than a few minutes, I guess. But if we had to repair a mutation, you'd also need a ligase to glue the DNA once you fix the mismatched base. All right, so there's your review of replication. Oh, it was literally a minute. Speaking of ligase, and all right, so let's say you get a mutation. This is showing what's called a thymine dimer. So thymine dimers will happen like if you're exposed to too much ultraviolet radiation, which is in a lot of sunlight. You will get, if you have, on a DNA strand, if you have two thymine nucleotides that are next to each other, they will form a covalent bond with one another, and that will create this little kink that pops out, where that's bad because that's essentially going to gum up the DNA replication machinery. It's going to cause some issues when we're doing protein synthesis, trying to transcribe that DNA. So we want to repair that kink, or if it's say this is a T, and let's say they put a C there or something when it should be an A, so this could, in other words, this could be a mutation. You will have an enzyme called a nuclease. So think of a nuclease as it's like they're like genetic scissors. They're similar to restriction enzymes when we're doing some DNA technology stuff. Similar idea. It would cut, and then DNA polymerase 1 would then come in and fill in the missing nucleotides. And then DNA ligase, that's the glue, ligase will glue those strands back together. Okay. Telomeres. So whenever we're copying, each of our chromosomes, we actually can't copy all the way to the end of the chromosomes. So what telomeres are representing would be these telomeres here in this like kind of orangey glow there. So the idea of the telomeres is like progressively the ends of these chromosomes will get shorter and shorter after each round of DNA replication. So basically, it's kind of an active area of research to try to figure out, into like extending life kind of research, like trying to figure out how can we prevent our chromosomes from getting shorter? How can you promote? There's an enzyme called telomerase that some species will have that will enable them to keep their telomeres from shortening. Some cancer cells will have telomerase activity, meaning their chromosomes won't get shorter, and that kind of almost makes like the cancer cells like immortal, quote unquote. So, anyways, just kind of something to be aware of, and that's kind of an active area of research. Okay, getting into protein synthesis. So that's really most of chapter 23 or chapter 13. So looking at protein synthesis, if we're looking at it in a bacterial cell. Bacteria are fairly simple in the sense that they'll do, just like we will, they'll take DNA, make mRNA, and that process is called transcription, and then they'll take that mRNA and through translation and the use of a ribosome, they make a polypeptide. I say it's simple or simpler because all of it happens in the cytoplasm. When I compare this to a eukaryotic cell where we have compartments, we have a nucleus, but anyways, just focusing on the bacteria, translation makes that polypeptide, and then this polypeptide, so you get one or more, so I'll say 1 plus polypeptides, will then equal a protein. Where this polypeptide, when it comes out of this ribosome, it's just going to be like a single, like what you see here, it's like a single, like a straight kind of string of beads. Each bead here represents an amino acid. Say AA for amino acid. That polypeptide needs to fold up into a three-dimensional structure. Here, notice with a prokaryotic cell, there is no alternative RNA splicing. Okay, so in other words, we don't have introns and exons. So jumping to comparing to a eukaryotic cell. Eukaryotic cell, this is what I mean by here we would have alternative RNA splicing. So that's what the alternative RNA splicing is, how you go from pre-mRNA to mRNA. Where in alternative RNA splicing, you would have these would be called introns. And then what's in red here, because it's part of the actual mRNA, these would be your exons. This would also be an intron. Okay. And the benefit of doing that is that's going to give us some more options for different you can basically one mRNA can code for many different proteins. I'll review that in a few slides though. Anyways, in a eukaryotic cell, we do transcription. Transcription occurs in the nucleus, so make sure you know the location of that. And then we have to transport that mRNA into the cytoplasm, where we'll do translation and make that polypeptide. So similar, translation's fairly similar in prokaryotic and eukaryotic cells. They both are happening in the cytoplasm. Okay, so getting into the details of, or well, really not even the details, just kind of like a little bit more on like overview of protein synthesis. DNA is organized into these genes. Each gene will code for a specific protein. Now, when we learn about the regulation of gene expression, I mentioned how like most of the DNA is actually not coding for proteins. But genes, if we just say it like gene will some way you can define a gene. If you define a gene as also including not just the literal sequences that make codons and make amino acids, if you also include what we call the non-coding DNA, that would help with regulation, then this would be more accurate. But anyways, when we're looking at DNA has two strands. One is the template strand. Now understand this, the template strand runs from 3' to 5'. Then you have a non-template, what we call a coding strand, that runs 5' to 3'. And the coding strand, they call it the coding strand because it is the same sequence with the exception of the U's and the T's as the mRNA. In other words, the mRNA is complementary to the template strand. We use the template strand as a template to make the complementary mRNA. Then every 3 base pairs, every 3 nucleotides on the RNA, we call a codon, and each codon will code for an amino acid. So then this would keep going on and on until you hit a stop codon, and that's how we'll know when we stop making that polypeptide. Okay, then how you know what amino acid you have. Like, for example, here we had the sequence like U, G, G, U, U, U. So we'll just stick with those 3 there. So like each of these would be a codon. How you know what amino acid they go with is with this this chart, where the first base is here, second there, third there. So you find your first base, U. So go here to U. Second base is G. So go till you hit the G column. And then now you're in this box here. And so find the third one, which is G. So UGG, that stands for the amino acid tryptophan. Now I want you to know that the very first codon is always AUG. And AUG codes for methionine, MET. I think of it as school starts in August, AUG, like we met in August. Okay, so make sure you know how to use a codon chart. All right, so getting into transcription. Well, if we want to start transcription, RNA polymerase must bind to something called a promoter. This whole thing, we call this a transcription unit. This would be basically the whole kind of sequence of DNA that we're going to use to make your mRNA. So first, you need to know the 3 phases of transcription. The first phase is initiation, where the RNA polymerase will bind to the promoter, and that will unwind the DNA. DNA gets unwind. We lay down our, this would be your RNA transcript, and this would be where we then start, moving along. RNA polymerase moves along, and we make this RNA in a 5' to 3' direction. So, important here, the template strand that the RNA is made from is going 3' to 5'. The mRNA transcript is 5' to 3', but the template is 3' to 5'. Okay? All right. Now then, so 3 phases of transcription. Second phase is elongation. So this RNA polymerase moves along the DNA strand here, and the RNA as the DNA appreciate here as the RNA polymerase is moving across the DNA will rewound. That's what they're trying to show here. And because the DNA rewinds, the RNA transcript will unpeel itself from the DNA. It doesn't just stay stuck to the DNA, and so we're done doing transcription. And then the third phase of all this is termination, where once we hit the terminating sequence, we would then have the completed RNA transcript. Okay. So getting into a little bit more detail on transcription and how we initiate it, think of the ta-ta box, okay? The ta-ta box, that is what the sequence that the transcription factors, I'll have a picture showing that. Really the previous pictures I've been showing you, I've been kind of leaving out a lot of details. Really there are transcription factors that will bind, they will look for this ta-ta box, and then they will be able to recruit and hold on to the RNA polymerase, and then RNA polymerase will be able to start at the start point and copy that DNA to make the mRNA. Ta-ta box, about 25 nucleotides upstream. So upstream would be this way, upstream relative to the starting point. Downstream is like going down into the actual coding DNA, the DNA that would be making the protein. Anyways, okay, yeah, here are your transcription factors. So, as I said in the last slide, it's a little more complicated than just RNA polymerase just binds and does its thing. You actually need to have other proteins that bind to the ta-ta box, these transcription factors. They then can recruit the RNA polymerase, and we actually call it RNA polymerase 2, to be specific. That'll bind to the transcription factors, and then you start getting that RNA transcript. And again, notice the 5', 3'. And keep in mind that we would have a helicase. There's a kind of a helicase-like function to some of these proteins that would help us to unwind it and get things going. Oh, and this when you're talking about the transcription factors plus the RNA polymerase, that whole thing, we can call it a transcription initiation complex, so all those proteins together. Okay, I mentioned how in eukaryotic cells, just eukaryotic cells, we need to process that RNA. So we have an enzyme called a spliceosome that will remove, so it's actually backwards. You would think that the introns stay in. Actually, the introns get cut out. So introns are portions of DNA that are actually not going to code for the protein we want to make. So how we cut those out is with the spliceosome, where the spliceosome is made of these small RNAs, and it's also made of some proteins. So this kind of background purplish blob, I guess like the bigger circle around the smaller circles, that is basically like a protein scaffold that's going to provide just like the structural support. But the actual, like where the actual action's happening is with these small RNAs. Where these small RNAs, they're going to bind complementary. So they bind complementarily, I guess you should say complementarily, to the intron DNA. In other words, if this intron DNA, let's say it was like A, C, G, then complementary to that would be U, G, C on the small RNAs of the spliceosome. Okay, and then that would cut out the intron. Okay, and then cut out all the introns you need, and then this would be your final RNA. So pre-mRNAs before the spliceosome done its stuff, then the mRNAs after the spliceosome done its business. Okay, so getting into kind of a bigger picture of, we're really just, I don't even think bigger pictures, right? It's just going over all the steps of protein synthesis. We have transcription, so transcription, make the RNA template. We do RNA processing where the spliceosome is going to remove those introns. So here is this is your pre-mRNA, spliceosome only happens in the eukaryotes. We make our mRNA. Now notice that there's something called a 5' cap and a 3' poly A tail. Okay. And these things have two functions that I want you to know. One is they protect the mRNA. They're going to protect that mRNA from hydrolytic enzymes. These are enzymes that will they turn polymers to monomers. So an mRNA is really just a big old polymer made of the individual nucleotide monomers. So the cap and the tail, they're going to protect it from that. Oh, with the cap, it is a guanine cap. So think of 5G. So that the cap has a guanine nucleotide. So first function is protects the mRNA from hydrolytic enzymes. And the second function, this is what you actually see here, is the cap and the tail will help us in transporting the RNA to the cytoplasm. Now then, kind of simultaneously happening with all of this is we have our amino acids that we ultimately want to string together to make the polypeptide. To get those amino acids linked together, they must first bind to something called a tRNA. So these are the amino acids, and this right here would be what we call tRNA, where the T stands for transfer. It is the transfer RNA. To be short, mRNA. Here's your mRNA. The mRNA stands for messenger. M and mRNA is the messenger RNA. Anyways, this tRNA has two binding sites. This top site, that is where the amino acid binds. This is where the amino acid would go. And then this bottom site, this is called the anti-codon. So we went over the codons, how you read the codon chart to find out what amino acid the codon codes for. So the question is, how do we get the proper amino acid to go to the right tRNA? Because tRNAs are specific to an amino acid. Okay, so you got 20 different amino acids, you have 20 different tRNAs. So the enzyme that's going to help, it's like a Match.com kind of enzyme that's going to make sure the right amino acid goes to the right tRNA. That's what this is right here. It's called amino acyl tRNA synthetase. So it's a big old enzyme that has a binding site for the amino acid and a binding site for the tRNA, and it's going to provide an environment where those two can be met together. And amino acyl tRNA synthetase, tRNA, easy for you to say. This is specific, just like the tRNA is. It is specific to an amino acid. Okay, so a lot going on there. Notice the location of all this is all happening in the cytoplasm. So, getting into translation, we have our mRNA that's undergone like the splicing and stuff. We have our tRNAs, and we call them charged tRNAs when they have their appropriate. This is a charged tRNA. It's got its appropriate amino acid. Now then, we will have our ribosome. The ribosome is the main thing doing translation, where the ribosome has two parts. It has a large part and a small part, a large subunit and a small subunit, where the small subunit is what's going to bind first. So the small subunit has an mRNA binding site that will bind to the mRNA. And once the small subunit has appropriately binded the mRNA, then the large subunit can hook on. When we're talking about ribosomes, they are the rRNA, or the lowercase r stands for ribosomal RNA. Ribosomes are also made of, they also have some protein too. And similar to like the spliceosome, the protein of the ribosome is really more of a peripheral kind of structural part of the ribosome. It's not really a catalytic part, we would say. It doesn't do catalysis. It doesn't really have an enzymatic function to it. It's just kind of there for structural support. Now, your ribosome is going to have these three binding sites. They are called the A, P, and E site. So think apes, where it starts in the A site. The A site is where the next amino acid binds. So the next amino acid goes to the A site. The P site, P for polypeptide. The P site is where you will have the growing polypeptide chain attached to a tRNA. And then the E site, the E stands for exit. It is the exit site where the tRNA that has delivered its amino acid will then be able to exit. So here's how this would work. We start in August, so your first amino acid is always methionine. So this would be methionine. So methionine would be your first amino acid. That's how we would start. Then the next amino acid would come in, and it would deliver, the next tRNA comes in, and it would deliver its amino acid. Now, in the P site is the tRNA that had previously delivered its amino acid, and it is holding the growing polypeptide. But this tRNA, it will want to eventually get to the E site. Think of like you're going through a car wash. It eventually wants to get out of the car wash. Once the tRNA that was previously holding this growing polypeptide has left, now the tRNA in the P site can transfer the growing polypeptide to the tRNA that's in the A site that had just transferred its amino acid to the growing polypeptide. So this growing polypeptide gets transferred to this tRNA. This tRNA in the P site moves to the E site, and then it can leave. And then the next tRNA, next charged tRNA comes in to the A site, and so on. The anticodon, we focus in on this a little bit more. Like here, this anticodon is AAA. Then again, that's complementary to the codon, which is UUU. Okay, and that's basically translation. Now, the last thing to talk about is, well, how do we stop this? How do we know when we're done doing this? Well, I mentioned the stop codons. So once you hit one of those stop codons, which is either UAG, UAA, or UGA, instead of a charged tRNA that's actually carrying an amino acid, you will have another molecule that comes in called a release factor that it and only it, excuse me, I get choked up when I talk about this stuff. It and only it will recognize the 3 stop codons. There isn't a tRNA with an amino acid that can recognize those stop codons. So it will bind there. And then we will have to use the energy of the GTP. So I don't care about the numbers, just do know that GTP is the energy source used here. Using the energy of GTP combined with the release factor, that will then cause dissociation. It'll cause the large and small subunit to break free from the mRNA, and then your polypeptide will then be free, and this polypeptide can then fold up into the 3-dimensional protein structure, and then that protein does its job, or maybe it pairs up with another polypeptide to make maybe a hemoglobin, like some sort of protein that has like multiple polypeptides. Okay, so another thing we need to talk about is how do we get this protein where it needs to go? And earlier in the year we talked about cytoplasmic and bound ribosomes. So those are the ribosomes that were bound to the endoplasmic reticulum, or we call it the rough endoplasmic reticulum, rough for like ribosomes. So like these would be ribosomes, they would kind of feel rough. Or actually these are nuclear pores, I'm sorry. But yeah, they're not really showing the ribosomes down there. Anyways, cytoplasmic ribosomes, these are for proteins that stay in the cytoplasm. So if you're a protein that's going to stay in the cytoplasm, you are made on a ribosome that only stays in the cytoplasm. Now, if you're a protein that needs to be transported out of the cell, so I should say exported. If you're a protein that needs to be exported out of the cell, or if you're a protein that's going to be part of the endomembrane system, you are going to need to be made on a ribosome that is on the rough ER. At least that's what I said to you earlier in the year. Now, it's more complicated than that because all ribosomes start in the cytoplasm. So I'm going to write that. All ribosomes start in cytoplasm. Meaning, when we're doing translation, all translation, or at least the beginning of all translation, happens in the cytoplasm. Where at some point, if this is, we're zooming in here on a protein that's going to be one of these two scenarios, exported or part of the endomembrane system, this protein, at some point, there's going to be a codon sequence that will code for a signal peptide. So that's the thing in green. They're a signal peptide. And this signal peptide will attract what we call a signal recognition particle. Okay? And we call it SRP for short. This signal recognition particle will bind to the green signal peptide, and it'll drag this ribosome and mRNA polypeptide, this whole thing, to the endoplasmic reticulum, to this little pore called the translocation complex. Did I call that a nuclear pore earlier? If I did, I'm sorry. I I've been talking too much today. I'm just going crazy. The translocation complex. This translocation complex, it translocates things. It translocates things from the cytoplasm to the inside of the ER. So what happens is the signal peptide, the signal recognition particle, binds to the translocation complex, and then as this is bound, we have paused translation. Once we are bound, we now resume translation. And that's what you see here. So we have resumed translation, and so now we are translating the rest of that, that mRNA sequence into a polypeptide. And until we hit the stop codon, then the protein would dissociate. You know, you'd have that the release factor binding into the ribosome, breaking everything apart, and then you get the protein in the ER lumen. And then that signal peptide right here, that green portion, would end up getting removed and discarded. It's actually not actually part of the functional protein. And then this protein can then move through the endomembrane system, go through the Golgi apparatus, and be sent wherever it needs to be sent. Okay, let me get some water. Okay, so this is talking about what's called a polyribosome. So let's say I want to make a lot of protein, or I just want to be efficient. I don't need to wait. We don't have to wait for one ribosome to finish doing its job before another ribosome can start. I compare it to like a water slide. You don't need to wait for one tube to go down the water slide before another tube can go down. You can have multiple ribosomes bonding to the mRNA at the same time. And that whole complex we call a polyribosome. Okay? This is something, this slide here, this only happens in bacteria, but this is called coupled transcription and translation. So the idea here is kind of interesting. Basically, because transcription and translation both happen in the cytoplasm of a bacteria can start doing translation with the ribosome before the RNA polymerase is done doing transcription. So in other words, as soon as the polymerase has gone past and made a portion of the mRNA, polyribosomes can form on that RNA transcript and start making polypeptides. Okay, getting into mutations. This is looking at an example of sickle cell anemia. So a mutation would happen where, here would be our normal, we call it wild type gene, where no mutations have happened. And let's say we have, this would be called a point mutation. It's a point mutation because you have changed just one single nucleotide pair. And let's say we change the nucleotide from T to an A. So now that would pair up with a T here. And the issue is, this is going to be your coding. This is your coding strand. So that's what becomes your mRNA, where T is swapped with U. The issue is now, instead of the GAG codon, you have the GUG codon, which codes for valine instead of glutamic acid. And that seems innocent enough, but that little tiny change is actually enough to turn a normal-looking concave red blood cell into a sickle-celled red blood cell. Specifically, a sickle, this beta globin will kind of end up having this weird sickle shape, giving the overall red blood cell a sickle shape. And that causes issues because that leads to more blood clotting and issues we talked about with sickle cell anemia. So getting into some other kinds of mutations. So up top here, we're looking at your normal wild type strand. Oh, something I haven't talked about with the protein is you always have an amino end and a carboxyl end. Okay? Where the amino end is at the 5' end and the carboxyl end is at the 3' end. So we will add each amino acid is added to the 3' end of the previous amino acid. Or another way of saying that is each amino acid is added to the carboxyl end of the previous amino acid. So looking at two different kinds of things going on here, we can have a nucleotide pair substitution like I showed you with sickle cell anemia. Here you're going to swap out that G with an A, which then will code for a T, which then makes a U, and then now what ends up happening is now we end up getting a stop codon. So we end up getting what's called a silent effect happening, where silent mutation is where you get no effect on the amino acid sequence. I'm sorry, I actually said that wrong. We don't by changing that T to a U, we don't actually code for a stop codon now. We actually will code for the same amino acid. So let me write this bigger. GGU, that would still code for glycine, as would GGC. If you go to your codon chart, GGU and GGC, those both code for the same amino acid. We call this third base wobble. Or just this idea there's redundancy in the amino acid code, where only the first two nucleotides are typically going to be essential for the amino acid. And for most amino acids, that third base can switch, and it will just cause no effect. That's why I call it a silent mutation. It's not actually going to affect the overall polypeptide. Now, this here is showing a nucleotide pair insertion. Now, insertions or deletions, they are going to have the biggest impact on that on the protein, and that's because of frame shifts. It's a really important idea to understand. By adding this extra A nucleotide in, that now is going to cause us to code for a stop codon. We call this a nonsense mutation. So nonsense, you now will code for a stop codon. Okay? But anyways, getting back to this idea of a frame shift, by adding in this extra A, we now the codon here is ATT, instead of, if you go up here it was TTC, now it's ATT instead of TTC. In other words, inserting or deleting a nucleotide will shift the entire frame. Everything after you insert or delete a nucleotide will now be messed up. That's why it's such a big deal. All right, going through these other examples, we can have what's called a missense mutation. So these things I'm writing here, understand that these can either apply to substitutions or insertion or deletions. Substitution, insertion, deletions, all of these can cause, these are the effects of substitutions, insertions, or deletions. Missense is when you get the wrong amino acid. So here we substitute a T instead of a C, and so that codes the amino acid serine instead of glycine. Here's an example of a missense. This would also be missense, but this would be a missense where we do a deletion. We delete an amino acid, and then again, what I was mentioning last time, as soon as you delete this, everything after this now gets messed up. So now all of the amino acids, because we shifted the frame, all of these amino acids are messed up. Here they're showing that this would be called a missense frame shift, because we are just getting wrong amino acids, plural, happening here. Okay. Last one, they're showing us a nonsense substitution. So here we substitute an A instead of a T, and that codes for a stop codon. And this is, we're deleting, okay, this is interesting. This is a 3, so if you have a 3 nucleotide, either insertion or deletion, they're bad, like they're not ideal, but they're not as bad. And that seems backwards. You think if you delete more letters or you add more letters, that would mess things up more. But you got to remember, codons come in triplets. They come in 3s. So by removing 3 letters, a TTC, I didn't mess up my frame shift. Not as bad because no frame shift. Okay? Meaning everything after that missing 3 nucleotides, or added 3 nucleotides, doesn't get messed up. We just end up getting, in this case, because we deleted, we end up getting a missing, this should have been lysine. We're just missing that. But then everything after the lysine, the phenylalanine and the glycine, that still preserves that original message. Okay? That's not saying the protein is going to be like fine and working properly. It's just not everything's going to be messed up. Okay, getting into chapter 15. So regulation of the enzyme activity. Well, yeah, I'm gonna stop this right here. This video is getting kind of long.